USA. His main research interests include non-linear PDEs, regularization methods, inverse and ill-posed problems, variational PDE-based image processing, and computer vision with applications in remote sensing, biomedical imaging domains. His current research focuses in data science and bioimage informatics with machine learning techniques. With this brief introduction, I hand over the session to Dr. Surya Prasad. Welcome, sir.
Yeah, thank you, ma'am. First of all, I'd like to thank Dr. Jayanti and Dr. Sudha for inviting me and present on this. Thank you. Present on this very important day. Today happens to be Ramanujan's 133rd birthday as well. So it has very good significance in terms of the mathematics. I'm going to talk about a little bit here. Today I'm going to hopefully showcase a few case studies in large-scale computational biomedical image processing and how we use strong mathematical models to solve some of these big data problems in biomedical image processing. So throughout my talk, I will have a unified theme of mathematical image analysis. So I will touch upon each of these techniques a little bit as we go along. The main bullet points here are like processing this biomedical imaging data. I will talk a little bit about some simple pre-processing steps to mid-level segmentation and classification models, and how do we analyze this biomedical images, and probably a little bit of visualization tools. But for the majority of my talk, I will be talking about how we process this biomedical imaging data. So obviously, there is an underlying theme of mathematical image analysis tools, and I will also talk a little bit about machine learning and deep learning without going into too much of the details. But as you know now, machine learning, especially deep learning models, are well utilized in the image processing area, and especially in the biomedical imaging context. So I do have a lot of slides, but many of them will contain images. So although my slide number says 100, I will try to stick to the time we have allotted. And please feel free to send your questions in the chat so I can reply in between too. So as I said, I'm going to talk a little bit about imaging informatics, but a lot of machine learning techniques is what we are currently using. So in this area, the main motivation is to create computer science systems that make sense to a biologist or a medical practitioner. The data in this case is trying to tell us what the answer is, but we just need to know how to listen to it. So I will, throughout the talk, I will mention some of the computational processes which try to mimic the biological process underlying the imaging data, and computational techniques we apply, such as the machine learning models I'm going to touch a little bit about. And not only that, not just applying these techniques, does it translatable, does it become useful to biologists and medical practitioners? So I arranged my talk in micro to macro scale. By that I mean, I'm going to talk a little bit about microscopy data, which is at the nanometer microscopy level, and then go one order of magnitude higher in terms of the spatial scales. So that's what I mean by micro to macro level imaging data. So I'll go from microscopy to histopathology to endoscopy to MRIs. And if time permits, I will talk a little bit about some computer vision problems, which are at even higher scale level. So let's head into microscopy-based image analysis. So as everybody knows, whoever is interested in science knows about microscopy. I mean, you have looked at, magnifying glass, things from your childhood, for example. So the microscopy is obviously very versatile and widely used across biomedical imaging domain, and undeniably very beautiful. You can, look at micro nano level scale now. I mean, now we are pushing on the optics so that we can even look at even much more smaller scale than a width of a human hair. So in this at least subtopic, I will talk a little bit about epifluorescence and confocal and optical fundoscopy images a little bit. But the recent progress has been into higher time-lapse imagery from lattice light sheet microscopy data, which I will probably not go into the details. But throughout this, the basic idea is to how do we use this microscopy imaging data and get insights. So as I said, computational microscopy is not a new area. The light microscope itself goes back to like 300 years old. Due to the humans are always wanted to look at the tiniest things possible, and recent computational and big data and machine learning models have paved the way for automatically analyzing this imaging data, not just look at them but store them and analyze and visualize. So computational microscopy area is where the imaging physics meets image processing. So we need high throughput and automated image processing pipelines. We need some very nice interfaces so that the biologists can not only image, but can also look at this automated image processing outputs. Excuse me. So as a case study, I'm going to talk about a project which my lab worked on a few years back, but we continue to work on it. These are known as dura mater vasculature image analysis from microscopy images. So without going into a lot of technical details of how the image is obtained, I thought it will be very easy to show visually because we are very usually very easily we can see through the human visual system. So the epifluorescence imagery is basically consists of staining some particular tissue, in this case a dura mater, which is a very fine tissue lining your skull and the top of your head. So this tissue has been stained with a particular biological agent so that it gives you a nice visualization of the blood vessels. So as humans, we can clearly see the blood vessels. Here we can in fact see the veins and very clearly popping out of the picture. But how do the computers can see this, right? So that's where the image processing tools are very important. So, but in terms of image processing, I've highlighted here a few of the problematic areas. So remember, the computer sees only the pixel values, right? Like it doesn't have the biological evolution we have had for millions of years. So in this case, the depth occlusion, obviously I'm showing a 2D picture of a 3D imaging data. So you can see that, there is a vessel going behind here and there is a bright vessel on top. So how do we identify this? And because of the way these images are taken, there's always a dye leakage. By that I mean the vessel may not be clear. It has diffuse boundaries, and it can also have this lumped clustered diffusion. And also this dye may not absorb in a uniform way. So what you see here in the binary red and blue maps are three physiologists who marked these images painstakingly in a Photoshop or GIMP or whatever your favorite Photoshop tool is. So they marked the boundaries and said, hey, here is the vessels and here is what I want you to segment from automatic algorithms. So in this case, the red and blue are the arteries and veins in this case. So for the computer, it's not that easy to segment, and we have come up with some nice pipelines to handle that problems I mentioned in the previous slide. So in this case, I'm showing again one of the pipelines we have tried. This is not the final pipeline, but as in any typical image processing pipeline, going from images to segmentation, you have kind of a plug and play architecture here. So you want to do some pre-processing, smoothing, deblurring, kind of very simple steps, super resolution to improve the resolution, and then apply some segmentation models. So I thought I will start with a little bit of simple pre-processing steps, which anybody who has taken a calculus course can understand. So again, as introduced in the start, I am a mathematician by training, so I have to have some equations, but not to scare you with the equations. These equations are actually very easy to understand if you know a little bit of image derivatives. Here, Ix is basically the x derivative of the image I, and Iy is the y derivative. So what we do is, in this case, we want to have a nice model which can fuse these images. So in this particular data set, we had what is known as single focus images. So we had the microscopists take the same image, same tissue at multiple locations in focus, and some of the focus areas are out of focus. So we had a varying number of input single focus images. So we wanted to first, can we combine this into a nice all focus image? So for that purpose, we again looked at a very intuitive technique. This is before deep learning and machine learning came over. So we looked at the derivatives. The derivatives usually tell you some edge information or focus area information. So in this case, what we did is a very simple technique. The technique is to assemble this IXX, IXY, IXY, IIY. This is known as a Hessian matrix of your image matrix. So this is a matrix for every, at every pixel, you can compute this matrix. Then you can compute the eigenvalues. So again, this lambda1, lambda2, you can compute at every pixel. So assume that you have n single focus images. We computed this quantity. So this quantity is known as the Frobenius norm. Sorry for jumping the slide. So this is basically the square root of the sum of eigenvalues. So we compute this, and we looked at which pixel this has the maximum value. So this again, if you visually see it, you will understand and appreciate this map better. So whenever this Hessian eigenvalues, or rather the Frobenius norm takes the maximum value, that is in focus. And if it's a lower value, it's not in focus. So you can choose a P from this set of images where that happens. So let me show this pictorially. So here, what I have is 6 different input images. The focus is varying from your top, rather bottom left to the top right. So we want to create an all focus super resolved image out of these 6 images. We want to create one master image where every area is in focus. So the multiscale Hessian Frobenius norm I mentioned here is plotted here as a color-coded schema. So there are 6 colors here, going from red to all the way to blue. So you can see that it is capturing from image 1, image 2, image 3, image 4, image 5, and image 6, where the focused area is. So now you go through pixel by pixel and then compute this fused image. So again, it's a very simple calculus-based technique, and surprisingly, it works really well. So I didn't yet talk about how we segment, but I just wanted to show you by fusing or even applying some simple pre-processing techniques, we can boost the further downstream image processing pipelines. So for this, we used a metric which is very widely known in image processing area, the dice similarity coefficient, which is basically an overlap between manual segmentation, things which we take it from 3 physiologists. We took a consensus. That's our set A, and automatic segmentation is B. I'll talk about the segmentation models in a bit, but I just want to show you just by fusing from, say, single optimal image to the fused master image, we got sometimes 122% of improvement in the segmentation results. So again, this can be appreciated if you look at the sorry for jumping again. Let me go back. So if you can appreciate this visually much better. So in this case, I'm showing the input single focus images on the top row, and the bottom row are all the corresponding segmentation methods. I've not mentioned yet what the segmentation model. Let's assume one of your favorite segmentation model is applied. Here, white means the segmentation model segmented correctly. Red means it actually missed from the ground truth. So here at the bottom is the fused image and the corresponding segmented image. Again, remember, red means you missed it from the segmentation model, and blue means you picked up. So basically false positives and false negatives. So here is the ground truth, which is drawn by 3 physiologists. So we took the intersection of 3 physiologists marked binary ground truth. So I can, as I said, a simple technique, which already works really well. So let me also give one more intuitive pre-processing technique which we have done. Again, we relied on simple gradient operations. In this case, U is your input image, and we smoothed with the Gaussian filter at different sigma values, and then we summed them up. So what it does is it captures gradients across scales. By that I mean you can capture multiple cell boundaries. So this also worked really well. As you can see here, even under noisy conditions, these are again blood vessels from a different data set. If you compute the eigenvalue of this assembled gradients, you get a nice clear edge map. So obviously this is going to improve your input image to any segmentation model. So we applied this filtering before doing the segmentation. Again, this is visually again showing with and without applying this simple pre-processing technique. Again, these are all before machine learning and deep learning models took over the world of image processing and biomedical image analysis. This is again, I'm going to skip this slide. It just shows at that time it was the state of the art in capturing, even broken vessel segments, which are which are very difficult to segment in this kind of cell blood vessel imaging data. So what we that was some few years ago, like 4 or 5 years ago, before the deep learning took over. So there are other kinds of image processing approaches. If you know you have enough ground truth or vessels, blood vessels marked by physiologists, then you can use training-based algorithms, like basically machine learning algorithms like SVM or random forest or other naive-based models. So in this case, we tested out benchmark different machine learning models. One of the difficult part is to get these images marked by physiologists. Obviously, it's a painstaking process. You have to ask them to draw these boundaries very closely, probably with sub-pixel accuracy. So what we had is actually a very limited training data set. So when we benchmarked this, we found that if you use random forest, which is a machine learning classifier model, it worked much better. Of course, in the random forest type model, it's a supervised learning model where you have to fine tune your features. So if you know certain features which work for your image, you should try to weight them higher. So in this case, in the blood vessel or fundoscopy or retinal imagery, it's well known that this kind of derivative filters and curvature, linear features or edge features will work well. So we tested different features and we came up on a set of 16 different features which worked really well in differentiating the results from the background. So the pipeline works as follows. We had close to 200 or 300 images, but we only had like 30 something ground truth images from 3 physiologists. So we had those training data and we computed this a set of 16 features, and then we let the random forest train on that data. And from now on, you can test on your test data, and we got good results. So originally, when we started, even with just 10 images, 10 RGB images of 1024 cross 1024, the accuracy was really high, close to 98. So this was a bit surprising, but not surprising in the sense of features, because in the retinal imaging world or in the blood vessel segmentation world, these are the very standard features people use all the time. So they are indeed, if you look at these features here a little bit closer, they're already capturing your vessels very clearly. So by fine-tuning this feature, we were able to get close to 99% accuracy on our data set. So here again, just to visually show and appreciate how this model works, what you see in the first row are the input images. You can see that there are a lot of problems with these images in terms of the background and diffuse vessels I talked about. Especially the first and the fourth image have very clear occlusions in the background. So obviously with kind of thresholding or simple image processing based segmentation, active contours or clustering models will pick up the background. So that's what you see, for example, in this Otsu thresholding, which is a very, very old model, and Niblack is another thresholding model. So these two rows here in the third and fourth row show thresholding models. So you can see that it picks up blues, false positives. It picks up a lot of background noise. And red means you're missing those pixels from the ground truth. So you can see the dice values here are not that high. And with random forest, you get always consistently minimum 80% or above accuracy, even in these very difficult, challenging images. So you can ask a question like, hey, why are you comparing with thresholding models, right? Like, so obviously we compared with other models, but in the interest of time and space, I'm not showing the results for other machine learning models here. So then the new deep learning wave hit us in around 2012. ImageNet came along and computing power became cheaper, and we were able to use very large neural network models. So we also applied. In this case, still remember, we don't have enough ground truth, but we still wanted to see how this neural network models perform. In this case, we used what is known as the VGG network, which is again a very standard network people use for the last 5-6 years now. So in this case, obviously the deep learning model will outperform even the random forest model. And one of the interesting thing I wanted to point out is we actually trained on ImageNet and tested on these blood vessels, and the accuracy was 94%. So even without having this training data, we were able to obtain really high accuracy. But if you use those 30 plus images we had, the accuracy shot up to 99%. So obviously these models are really, really powerful. One of the bigger advantage of deep learning models is, remember in the previous slide of um, random forest, I talked about this hand-tuned derivative-based or ledge-based, curvature-based features. You don't need to do any of that in the deep learning space. You just let the neural network take over, compute all kinds of features within the neural network model. But again, that's also a criticism of deep learning models, that you don't know what it is learning. Is it overfitting, or is it learning features which are not useful? But in recent times, there are now explainable deep learning models where we can prove indeed that it is learning the blood vessels, not the surrounding areas. So in this case, we obtained state-of-the-art accuracy. So here is a benchmarking result. Again, this is just a snapshot of the comparisons we have done. So OTSU is basically a very simple, lame thresholding model. This is a line, MS line is a line-based direction model, which is popular in retinal imagery. DBNet and DBNet2 are again two different network models, which we based our VGGNet on. So just to wrap up this subpart of microscopy imaging, image processing, again, I showed only a few of the parts of this plug and play architecture, starting from fusion to filtering and segmentation. I kind of went through fast in terms of the segmentation models. We also applied post processing and network extraction. So let me talk a little bit about that. What's the main advantages of why do we worry about segmentation accuracy? The reason is we want to extract some physiological properties out of these blood vessels. So these blood vessels have certain physiological hypothesis behind. When these blood vessels curve a lot, you have hypertension or some disease. So what we wanted to do is, after segmenting, we want to compute what is known as the medial axis and compute this angles and curvatures and tortuosity of these vessels to see if you can quantify how the change happens, say between between normal patients versus abnormal patients, or between a normal mouse or an abnormal mouse. So we did that. Again, in the interest of time, I will not go into the statistical analysis part, but the image processing part is very important, because unless you get a good segmentation, you cannot extract all of these physiological parameters. So that's why I want to emphasize that the reason why we want to push the segmentation state of the art is to get all the vessels possible without getting the background. Another advantage of developing such plug and play architecture is that you can somewhat take the same pipeline and apply to other imagery. So in this case, I'm showing what is known as human epithelial cells. These are not like the blood vessels. These are not curvilinear objects. These are more like blob structures. But you can try similar approach, going from fusion to denoising to segmentation to classifying into foreground and background. And that's what we did. Even if you do out of the box, by the time you don't need to modify any of that, just use the same pipelines, we still got good accuracy. Of course, if you fine tune your machine learning model based on the features for example, the curvilinear features will not work in this case because you have blob structures. So people usually use some other features here. So we used those features and apply random forest again. It was state of the art around 2016. But of course, now deep learning model is much higher accuracy than random forest model. But around 2016, this was like state of the art model there. So hopefully, I've convinced you some plug and play architecture can be useful in microscopy image analysis. We talked a little bit about multifocus fusion and filtering, touched a little bit about segmentation. I skipped over the statistical part. Obviously, this is extensible to other vasculature images, right? So these features in random forest or a deep learning model can be applied to angiograms or retinal fundoscopy or cryo-EM and neural imagery, confocal photon microscopy, etc. There are other extensions possible. I only showed 2D imaging, 2D image segmentation, but we have done 3D and 3D plus time. So in the interest of time, I will not talk about that, but I'll be very happy to take any questions you have about this microscopy image analysis. So you do all these image segmentation models and prove that you're better than others, but is it going to be useful? So one of the bigger thing usually lacking in image processing literature is, can it be apply it to nice visualization where the biologist or medical practitioners can easily use it. So we have done some work on towards that in terms of developing a cloud-based and local installation-based visualization tool. Again, this is kind of 2 or 3 years old work, but now we have deep learning techniques plugged in here. So the biologists and medical practitioners they do not care about the gory details of how you develop the algorithm in the background. They just want to upload the image, click, let an algorithm run, and give the segmentation. So it's also very important when you develop a biomedical image processing algorithm to be aware of how useful it will be to the users. So we found a lot of problems in applying standard image processing and putting it into a nice software visualization tool. We found that they do not scale well sometimes. They do not have the properties which the users like. So we adopted 2 or 3 of the previous models I've talked about, and those are available for the physiologists. So this we used as a constant feedback loop, and we got more ground truth data by let them mark and semi-automatically run the random forest model or a deep learning model and get more segmentations out. So that's also very important in terms of developing biomedical image analysis pipelines. So I will shift a little bit into a higher scale. I'm going to talk about what is known as histopathology images. And I will touch 2 application areas, so breast cancer and brain glioma. So in this case, this computational pathology actually is a very famous area now because all the pathology images used to be in glass slides, but now everything is computerized. So we have tons and tons of computerized pathology imaging. So in this case, it's a biopsy taken and cut into thin pieces and again stained with some, in this case HNE, which is a particular stain, a commonly used stain in histopathology. And these stains are scanned and stored as digital slides. Now, you can imagine that these slides are comparatively bigger, sometimes in gigabytes per slide, per patient. So again, the same problem of image processing arise here. So computational pathology is where pathology meets the image processing pipelines. The one strategy is to identify abnormal tissues, abnormal regions in these tissues, and apply segmentation models, and also obviously plug into a pathologist pipeline so that he or she can click and do this nucleus segmentation or stroma epithelial segmentation and classification. So again, I'll give a brief overview of a project we have conducted in breast cancer. So in this case, these are breast cancer biopsy tissues. The breast cancer is one of the common cancer among women. So the pathologists look at these slides and look at certain properties of this imaging. So they look at contextual relational features like how are these nuclei are separated, how are the spatial location between the stromal matrix in the background and epithelial cells in the foreground. And what we want to do is have a automatic classifying or image processing model which can do the same automatically across patient cohorts. So there was a paper in 2011 which kind of changed the computational pathology world. They proved that machine learning models can predict as good as a pathologist. So this was a breakthrough paper. So we looked at and we borrowed some of the ideas from this paper from 2011. So we looked at certain properties of these images. So we looked at standard ratio between epithelial and background stromal cells, and we looked at a bunch of features. So in the original paper, they used a certain number of features, and for the breast cancer data, because of the way images vary between sites, so the original study was from Stanford, and our study was from Missouri. So we have to modify some of these features. You can prove that those features actually help in predicting the survival of the breast cancer patients. So in this case, we had the Stanford data as well as a couple of other data from different patient cohorts from Europe. In this talk, I'm going to restrict myself to image analysis part. I will not go into the CAD and survival analysis part. So here is our plug and play pipeline. So it's again very similar to microscopy. plug and play pipeline I talked about. So we have level 1 of pre-processing, low-level image processing stuff, and then segmentation, and then higher level classification models. So this is again a kind of an old work where we used a simple octave contour model. I'm going to skip these slides because there's a lot of math here, but just show the intuition of it. So what we did originally is to split the image. Obviously, these are H&E stained color images. So we thought, oh, okay, maybe we can easily split into pure brightness and chromaticity. Chromaticity is the pure color information. So C1, C2, C3 are like red, green, and blue channels. So obviously higher grayscale value means it's higher in the red channel or green channel or blue channel. So we split that into two, and then we applied this a particular active contour model. So FD and FC are your feature fitting terms, and this gradient of U tells you how the contour should move. So again, let me show the picture so that it will become much clearer. So what we did is we devised an active contour model, so which is like a rubber band moving across your image, and it'll try to catch on to the boundaries. So to aid the catching mechanism, we used chromaticity and brightness decomposition. So we split it into we split the image into B and C. So the vector C is actually 3 images. So we split the 3-channel image into a 4-channel image. So obviously we are increasing information here. And this is the feature we computed. So this feature is again based on some simple gradient formulations. And this feature obviously helped the active contour to stop on the boundary. So now you can easily see that the nuclei is captured and the background is in the outside of the active contour. So this gave us very accurate nuclear segmentations. All right. So this was a very fast scheme. So again, I skipped the equations. So let me go back a little bit. So what we do here is this minimization model. So we can prove that it's a convex minimization. And if you know a little bit of optimization, convex minimizations are really, really fast. So you can put it onto GPU and you can actually solve them very fast. So that's exactly what we did. Original implementation was like less than a second on a standard image size. But remember, in this histopathology, you have like 10K or even 30K cross 30K size images. So again, without going into all the nuts and bolts of the model, let me just show you how it works in real data. So this is from the Stanford breast cancer patient cohort. So what you see here, input image and the ground truth. In this case, this is actually not a nuclear segmentation application. Here we want to segment a clump of epithelia and as stroma. So epithelia corresponds to your nuclei. So the red regions are your nuclei, which are clumped together. So this brightness and chromaticity gives you some information about where they are clumped together. So on the E and F columns are the segmentation output before applying some post-processing techniques. So you can already see that it's pretty good for such a simple model. You can compare and quantify this using again the dice coefficient because we have the ground truth. This table just shows you how much between nuclei and the background, how much accuracy we got with such a simple ground truth, which is such a simple active contour model. But again, by the time deep learning came, we have switched over to the much more powerful machine learning deep learning models. So in this case, again, I cannot go into the exact details, but this gives you a simple overview of how we use the deep learning model. So we basically used a convolutional neural network model, which has stacks of convolutional filters and layers. And our network actually worked much better than the active contour models, obviously. And obviously, scaling up is an issue in terms of deep learning models. But now, currently, there are efforts in making this work on GPUs so that you can run for even whole slide images, which are like 30K cross 30K images. And again, visually, I'm showing a comparison of a pathologist who marked certain regions. And here it's a one layer of max pooling from the neural network model and the final output here at the last column. So we are currently working on multiple extensions of this. So I only showed a two-class classification problem, either nuclei or a epithelia clump together. But you can think about multiple classes, and you can also think about using these features, to other cell types, and you can also you know, see how it can be incorporated into the pathologist pipeline. So they use a particular system. Can you have these algorithms plugged inside? Okay, so that's about histopathology and a very similar plug and play architecture, similar to microscopy. So I'm going to go a little bit higher scale here, go to very nice imaging modality. Many of you may not even be aware of this, but it's getting very popular for gastrointestinal exams. So this is known as wireless capsule endoscopy. So this was a project I started in 2011, and my lab continuously doing it. This is a very interesting imaging modality. These are the capsules which have a very tiny imaging system, and they take images of your gastrointestinal tract. So you swallow this capsule and it starts taking pictures of your GI tract, and it's a lot of data. Now with the advantages of technology, you can go up to 35 frames per second and 8 hours of video. So this is now being widely used also in India nowadays to look for abnormalities in your gastrointestinal tract. So this is how typically your small bowel and esophagus and colon look like, and these are the capsules. Actually, these are the previous generation capsules. Now much better, much more powerful capsules are available in the market. Here is again a state of the art up to 2016. So these capsules are very tiny. It's like a small tablet, and they have either one side or both sides cameras, and has LED flashlights. So it flashes the light and takes a picture and conserves the battery. So, but in terms of visualization, the gastroenterologists are very, very happy because they can, they can see this image and say, hey, here is a bleeding, or here is, the abnormality, or here is a polyp, or here is outgrowth. Then they can do follow-up studies. But in terms of image processing, or people like us, or people like me who do image processing on this, like post-processing, it's super difficult, primarily because these are multiple lights and your imaging system is close to the surface you're imaging. So this is not like, taking a photograph, in your room or in your outdoors. So this is something known as perspective projection model. So the lights are very close to the object. And because of the way it is conceived, the intensity saturation is a very big problem. So the light flashes a very high intensity value onto the surface. So some of the areas will be like completely washed out. Also, it's a huge data. So per patient, you may have like close to 50 to 60,000 frames to analyze. And another bigger problem is because of the way it's devised in terms of, patient should not have any discomfort. So just follow it and forget it. And it sends the images to the belt and it's uncontrolled motion. So sometimes the capsule may get stuck in one area and give you thousands of images of the same place. So obviously, we started with some of the simple problems to attack first. In this case, what you're seeing is your colon in your gastrointestinal tract. This is the center part is where the capsule is moving. The gastroenterologists want to have a very simple scheme which separates this tissue from the center part. But even this simple problem is a very difficult task for automatic image processing because it's there's a lot of bubbles and trash and illumination problems and the washouts I talked about. So we had a pilot project on this. So we came up with a simple kind of an active contour model I introduced in the previous histopathology area. We tested it out. It works okay. And we compared again with simple Otsu or mean shift clustering and active contour models. Here is another example of how the segmentation works. So the white boundary is the segmentation output. So here is again another example. Okay, this video will not play here. I'll show at the end of the talk, if time permits, the video of the segmentation. So another important task in this imagery is detecting these abnormal growths. These are known as polyps. So this can be precursors to colon cancer, and can we identify these polyps from this video data. As you can imagine, it's a needle in a haystack problem. It occurs only in few frames out of the 8-hour video. So usually the gastroenterologist looks at their screen and go through the whole video, and it takes for them 2 hours to find out where it is. So you can already think of how image processing can help here. Can an image processing algorithm go through the video and flag, okay, here is probably a polyp, you need to take a look. So again, this is before deep learning models came through. So what I'm showing is a very simple model based on calculus. So if you know a little bit of calculus, or if you have done calculus 101 in your engineering course, the curvature of a sphere, it's 1 by r. So we used the same technique. We thought, okay, this polyps and your colon is like a cylinder, and the polyp is protruding. So we wanted to measure how much it's protruding. And of course, it's a very idealized model. Polyps are not going to be that much, circular. But let me go back a little bit. Oops, sorry. So here are some examples, right? Like there are some pedunculed polyps, meaning it's like a mushroom, and some of them do not have any stem. So we want to see how much it's protruding in this tube. So think of your colon as a tube, and this is protruding. So we devised a very simple curvature-based protrusion amount. This anybody can implement. Even in MATLAB, it's like 3 lines of code. So we computed mean curvature and Gaussian curvature, and if you play around with it, you'll see that this particular quantity P, which is basically minus minimum between mean multiplied by the Gaussian, will give you the protrusions, not the convex object. It will give you the concave objects. So as you can see here in the D, in the synthetic case, if there is a protruding sphere, you'll capture it. And also, if you know a little bit of curvature, it's all constant value here. So the orange region is same value. So that value of P should tell you how much protrusion is there. Again, not all protrusions are going to be polyps, right? So that's exactly what we found out. So before we apply and compute this P, we did some pre-processing. We basically took the images and smoothed completely so that only the protrusions will show up. And again, this table is not intuitive. I'll show in the next slide the images. When we cut off these protrusion values, we were able to find out polyps very fast. So let me show you what I mean by the protrusion amount. So here is the input image, and here is the protrusion value. So you can see that P above 3000 will tell you where the protrusion is happening. Why is this useful? Because in polyp frames, we did not miss any of it. We captured. And in the no polyp frames, obviously the value is so low, we can obviously avoid these false positives because there are no protrusions here. So remember, in this kind of imagery where you have 8 hours of video and the gastroenterologist is looking at already 2 hours, you want to reduce the time to 30 minutes or something. Obviously, you cannot reduce like, here is the polyp frame. That's going to be very hard, even for deep learning models. So in this case, our aim was not to have a high accurate model, but rather have less number of false positives and do not miss the true positives. So we reduced from 55,000 frames to close to like 1,000 frames, which is already a big time gain. So that's the reason why we used a very simple protrusion measure, and it worked, and it was applied on the field, and we field tested it, and we were able to find not all the polyps, but almost all the majority of the polyps. So let me talk a little bit about another problem in this area, where the gastroenterologists want to view them in kind of a 3D model. So remember, we are very comfortable with 3D world, right? Like our world is full of 3D objects. So we want to have a 3D visualization of everything. So in this case, the images I've shown are all in 2D. So there was a nice approach called shape from shading from computer vision. We borrowed the idea, and we were able to show from 2D to 3D for this endoscopy images. So this idea is nothing new. It's been there for the last 30 years. And what it does is, given a 2D image, it tries to plot the 3D shape of it. So it's called SFS, or shape from shading. It's again based on simple calculus. It works based on the image irradiance equation, because images are obtained based on how much irradiance there are. And if you know where your light source is, you can actually reconstruct the 3D shape. So again, there is a minimization going on here. I will not go into the details. Here are some examples of our imaging model. Remember I said in the capsule endoscopy, the lights are very close to the surface. So you have to precisely geometrically model that. That was done by this function here, or n is your number of lights. And you minimize this whole thing and compute the z. Z is your depth map. So here we modified the shape from shading for endoscopy images with the model I showed in the previous slide. And this shows, these are synthetic images, and this shows how much error we get. So here are some, again, examples of how that shape from shading model works for real data. This was really interesting because we benchmarked this with gastroenterologists, and they were very happy because now they can rotate and see things, or zoom in and zoom out, and interact with 3D models. So it captures all the mucosa folds, which is basically the tissue, and it doesn't capture liquid material, which was an advantageous thing because they can now zoom into only on the mucosa area. So here are some more examples of different structures of endoscopy images. So this also can help in identifying how big the polyp is, because now you can draw, you can do scale measurements as shown in the final figure there, which is really useful for the colon exam. This is applied to non-capsule endoscopy. So this is a standard colonoscopy. This is not a wireless capsule. It's a standard wired colonoscopy device. And we proved that at the time it was the state-of-the-art model there. This is again on a standard colonoscopy image, how the 3D imaging looks. So you go from 2D to 3D, and you can clearly see the structures on the polyp and the mucosa. Here are some more examples of different structures gastroenterologists are very interested in. You can also look at the bleeding region much more close. Polyps and peduncle polyps, or the surface of the polyp, you can visualize much better. So just to recap, hopefully I've shown some glimpses of image processing in the endoscopy world, like mucosa or tissue structure detection. Also, a very simple geometry-based polyp detection. It's a protrusion indicator. People now use that to go from full video to particular focused regions of the videos. And then you can apply your machine learning and classification models to find out the polyp and polyp size and classify them into different classes. I also showed a little bit of 3D visualization models we can create from these images. And with that, I will again jump scales and go a little bit higher, which is MRI and CT and PET. These are again very well-known imaging modalities, and almost everyone is aware of these imaging modalities. So here, again, I'm going to restrict to neuroimaging. Obviously, there are liver MRIs and liver ultrasound my lab works on, but I thought I will share only neuroimaging image processing pipelines. Here, radiology meets image processing. We need data-driven approaches, and we need automated image processing pipelines. Here, we really need 2D and 3D image processing because the brain is in 3D, and you want to apply automatic image processing models on 3D. There's also various modalities. So I'm going to only talk about CT and MRI, but obviously there's fMRI and other SPECT and PET imaging, which I will not talk about. And also, how do you use years and years of radiologists' expertise? So let me show you a very simple task here is to segment brain MRI, different regions of the brain MRI, like white matter and gray matter and the ventricles. So here what you see are three different cases in terms of the noise and the intensity inhomogeneity from the imaging scanners. So what you see on the left are input images, and what you see on the right are a particular segmentation model, which works and captures 4 different regions. So in this case, again, this is before deep learning models. So we used one of the state-of-the-art active contour model, which I will not go into the details. But again, it puts contours around objects of interest. So what you see here are input and gray matter on the background, and I'm showing only the final segmentations here. And you can show that by segmenting with highly accurate models, uh, you can show the histograms to show did you really segment uh, these 4 distinct regions. So these are histograms showing for different noise levels and intensity and homogeneity. So again, this can be used in a plug and play architecture. So this is currently being used in one of the centers here. I only showed filtering and segmentation model, but there are visualization tools where we plug in these models too. So we go from filtering to segmenting to visualization. There are other very interesting problems in neuroimaging, especially in MRI data. Before even you apply segmentation, you can apply skull stripping. Basically, you remove the skull before you apply segmentation models. In also neuroimaging, one of the important problem is how much deviation the brain has from the standard normal brains. So this is known as mid-sagittal plane detection. And again, without going into the models here, I just want to show pre-processing steps we use. This is denoising model we use for removing noise from P1 weighted and T2 weighted and phase contrast images. So these are not neuroimaging, these are just standard cardiac imaging. Here is the neuroimaging data. This is the corpus callosum showing how the model is working. Obviously, I only talked about MRI, but in the neuroimaging community, people have now more modalities. So how to combine them and register them are also very important problems. So we are working on how to combine CT and MRI for neuroimaging. With the current deep learning models also, we are exploring how we can do certain tasks. So in this case, I'm showing detecting brain hemorrhages. Again, this is simple basic models from neural network community, VGG and Inception and ResNet. Even if you just take an ensemble model without even modifying anything, you can go up to 94. So which is very promising. Now we can take these models and modify and see if we can push the accuracy better. So we are currently applying this for ICH, which is intracranial hemorrhage and bleeding detection in the brain. Like I said, neuroimaging has a lot of other imaging modalities. You can think of denoising and classification here as well. There are image fusion problems you can think of where you can combine 2 different imaging modalities. Here it's CT and PET. So to cap this particular imaging modality and neuroimaging, here again, filtering, denoising, segmentation are important. Here the noise models are completely different from microscopy, for example. And you can also think about multimodal fusion models, and we want to obtain state-of-the-art results here. Okay, so what are we thinking in this area? So in this area, we are thinking going beyond 2D to 3D and also looking at tissue classification and tractography of brain structures. Obviously, I don't have time to go into all the deep learning models, but we are currently using deep learning models almost in every project I've talked about. All right, so let me give an overall summary of what I've talked about today. Hopefully, I gave you snapshots of different image processing techniques from filtering to fusion to segmentation. I touched a little bit about machine learning and deep learning because of that has to be a separate talk on how we apply those models. And also, I want to stress the point that all of these projects involve a two-way feedback mechanism with collaborators. So we develop algorithms, but we benchmark with the real, whether it's gastroenterologists or pathologists or radiologists or microscopists to make diagnosis and decision making easier. We are not going to replace all these human experts, but rather augment them in a computer-aided diagnosis system. So hopefully I've given you a motivation of why we need to keep developing these computational biomedical image analysis tools. So similar techniques can be used in computer vision. So these are a couple of projects in our lab we used to do, but now we do a lot of biomedical, where we used it for similar to shape from shading. I talked about endoscopy, but think of like aerial imaging scenario where you want to apply shape from motion because there is a camera which is going around your city and taking photographs. Can we reconstruct the 3D model of your city? And there is a technique called shape from motion. Again, here deep learning models are higher performing than standard geometrical models. So this is again very similar to shape from shading, but a little bit higher order complexity because now you have more images at going around. So reconstructing this 3D is known as shape from motion. And I talked a little bit about cell segmentation in microscopy area. So remember, detecting cars in images is somewhat similar to cell segmentation, right? So these are cells in blood cells or epithelial cells I talked about. Cars are like cells. So you just track them and segment them. So you can already see how techniques from standard image processing can be used in biomedical and vice versa. So my lab also works on other associated image processing problems, but obviously with a lot of biomedical application now. So I will stop here because I want to take some questions if you have any. If not, I will switch over to another slide deck. I want to talk about a little bit about a couple of very focused projects. If there are no questions, I will switch over to the next slide. Right. Okay, so let me switch over and so one more. Okay, before I switch over to the slides, here's a shameless plug for my lab. Let's see if we can put it in the full screen mode. So as I've shown in the previous presentation, we work on a variety of imaging informatics projects from radiology to neuroscience. We use fusion segmentation models I talked about. We use machine learning based classifiers, and we apply them for computer assisted diagnosis models. And these are some of the current projects. Obviously, with the deep learning models, we switched completely from classical machine learning models to deep learning models now. Please feel free to drop me an email if you are interested in any of these projects or if you have further questions. All right, so let me go to a couple of other interesting projects I wanted to talk about. Okay. Okay. Let's see. Screen again. All right. So I'm going to talk about. Okay, I talked about my lab already, so I'm going to skip these slides. So I thought, I talked a little bit about, machine learning and deep learning. So here are some thoughts from a very junior machine learning guy on example projects. When you talk about deep learning models, right, like there's a lot of hype now. It's very hard to predict where we will be in terms of deep learning models, and does AI is going to take over all the biomedical, world. Forget about the normal natural imaging world, but biomedical imaging world. What I'm going to advocate is the careful usage of machine learning tools. So I come from a different background, and now I'm working in a place where we are very resource constrained. We are not Google or Facebook or big companies where we can spend endless amount of money and create big AI models. So the world is a very resource constrained place. We want to apply machine learning models which work on a very resource constrained manner. And also, it's very hard to predict, right? Like people are talking about, AI taking over the world. My thoughts are like, it will not be, at least not in our generation. So here are some historical anecdotes, like people trying to predict future, which is always a very hard thing to do. So there is a lot of hype cycle for AI. So here is a nice figure showing how deep learning has been hyped. It is certainly really, really useful than standard machine learning models and image processing models, but it's really, really hyped. So I want to emphasize a couple of other ideas which came about, generative models which generate synthetic data, but is it hyper real? So remember, deep learning is a subset of supervised learning models, right? Like, so when people say AI and deep learning is only a sub part of the whole AI domain. So in the biomedical area, when we want to use machine learning models, we have to be really careful. We have to, without careful implementation and taking care of the bias, how much training data you have, are you really detecting or are you overfitting? Your algorithm may be giving very wrong answers. So here is a very nice example from a recent paper which appeared last year in Science. This is based on what is known as GAN, Generative Adversarial Network. What they shown is, GAN can generate synthetic images, real-looking synthetic images. This is very concerning for biomedical image processing, right? Because you have this notion that deep learning will classify, say, the skin cancer, for example. Like I showed in my previous presentation, it indeed obtains like close to 99% accuracy. That's really great. But what if I perturb the image a little bit? So that's exactly what they did. They added an adversarial noise, which is after adding, the image looks very visually similar. So great. So the algorithm should again find the same skin cancer, right? Apparently not. So when we apply a CNN model to this, it says, going from benign skin cancer to the malignant skin cancer category. So this is very dangerous because now, even if with a simple perturbation, the complete diagnosis changes. So in this case, how a medical practitioner is going to trust your algorithm, right? So this is not just an imaging domain. It can happen also in the text NLP domain. Here's an example they have added, right? Like they added a simple text substitution, and the final result was like, abuse risk was completely changed from high to low, and the insurance was denied. So this is a very big problem, right? Like, how do you make sure? So now there are a lot of models which incorporate this adversarial attacks to make better models. Um, I want to show a couple of projects we worked on. So I talked a little bit about histopathology for breast cancer. We are currently working on brain cancer, brain glioma. In this case, I just want to show, again, I'm breezing through this, but I'll be very happy to take questions afterwards. So this case, we want to look at this pathology images and categorize glioma disease stages. So this is known as automatic disease stage classification, and pathologists do this all the time. They look at your tissue biopsy and go, okay, you have grade 1, grade 2, grade 3, grade 4. And obviously the higher grade is severe brain tumor. In this example, again, visually I'm showing GBM versus LGG. I'm not a pathologist, so I cannot find out which is low and high. Obviously, GBM is higher than LGG. L stands for low grade glioma and glioblastoma. But how do the computers can do this? So we again went through the machine learning route, deep learning route. We obtained these images, and we did some pre-processing to avoid patches which are not useful. We did some pre-processing to convert and segment. We again went through similar machine learning models before going to deep learning. We looked at features people use in the computational pathology literature, like nuclei, structures, and whatnot. When we tried standard machine learning models, we got like good accuracy as well, but spatial features kind of dragged down. So what we came up with is a kind of a neural network model, and taking into account all the problems I talked about. Like if you add a little bit of noise to these images, what happens? So adversarially robust neural network model is what we have started using now. And here are standard neural network models people use. So this area exploded into 2012. And now these backbone models are available in any standard language you can imagine, Python or PyTorch, TensorFlow. And so you can easily adopt them. So we use that to have a nice classification model now, which is really encouraging. So here is, again, preliminary results comparing to classification accuracy into based on which base model we used from deep learning world. Um, we are currently coming up with our own optimized pipeline, which can use this adversarial attack so that it avoids predicting wrongly. As you can imagine, it's a very difficult problem. Without adversarial attacks, obviously the classification accuracies are really higher. Again, due to lack of time, I'm going to skip that. And let me see. I'll skip this as well and go to another very interesting problem. So in this problem, we want to analyze patients' activity. Again, this is a very well-known area in computer vision where people have models to predict what you are doing from videos: walking, jogging, running, boxing. So these are the data sets available in the computer vision literature. And the traditional computer vision models look at spatiotemporal feature detectors and have some classifiers to say you are jogging, running, or boxing. A typical analysis pipeline would involve extracting these features and applying some classifier to predict. But now, deep learning models have taken over, and they use convolutional neural networks or LSTM models to predict and track the trajectory. That's because there's a lot of data available now. So we have these important data sets which are can be used for your training data. The problem which we are concentrating now is what is known as human pose estimation. So from the video, we want to identify the human pose. So we are currently studying these deep learning models to see how the pose changes in the video. Also, we want to do this without extra setup from standard cell phone cameras and standard videos. There's a lot of literature on using depth sensors. We want to avoid that, and we want to see if you can predict your pose change across time. Okay, I am breezing through here, but the ultimate aim is to take 2 videos, compute this pose, and see how much deviation happened. Let me skip these slides and show the final slide. So there are some very nice models from Google and Facebook now on identifying pose real time. So we adopt these models to compute the pose, and you can try these demos which are available online. I'm going to skip that and go to the last slide to kind of unify both the talks. So we want to apply AI or machine learning models now. Having said all the drawbacks, hopefully we will avoid as we go along to applications across human lifespan. So there's a lot of biomedical imagery, from images I've showed in the previous talk, and there's also other types of data, variables and text and language. So my talk in the previous slides are all about imaging, but you can imagine similar problems in other domains. So here is a nice overview, like from Eric Topal in Nature Medicine. He predicts that AI models will take over at every stage of life. So my hope is also that when we develop this image processing and machine learning based models, we hopefully solve human life's every stage problem as we go along. So I will stop here, and if you have any questions, I'll be very happy to take. Thank you. Okay, I do see one question. How can we determine the number of hidden layers for classification? Okay, that's an excellent question. So that's primarily dependent on your problem. So now with the deep learning networks, right, like neural network models, one simple rule is it depends on the dimensionality of your problem. So there is a TensorFlow playground. If you just Google TensorFlow playground, you'll see a simple browser-based explanation of neural network model. So you can try playing around with the number of layers. And if you change the number of layers, it may or it may not classify into 2 classes. So I highly suggest you try that to get an intuition. So there are intuitive rules, but there is no hard fixed rule on how many layers will help to do your classification task. So it's highly problem dependent. So that's the short answer.
Thank you.
Yeah.
Thank you.
Okay, any other questions? Probably I went too fast. So okay. All right. So if you're, if you have any questions, please feel free to reach out to me. I'll be happy to share the slides as well, and um, you know, have some answers for your questions if you have any afterwards. Okay.
Participants, do anybody have any other questions?
Feel free to ask him, participants.
Yeah, probably I went too fast. So I thought I'll give kind of a plethora of biomedical image processing problems. Yeah. So if you do not have any questions, it's okay. But feel free to reach out if you get any related doubts or questions in this very exciting area.
Sir, one more question.
Sure.
Sir, I want to know the difference between the microscopic image and electron microscopic image.
Yeah. So I do not work on electron microscopy data, but there are a lot of different models now in electron microscopy data where it's much more complicated in the sense. So I showed only like blood vessel based microscopy data. So we have other microscopy data like confocal immunofluorescence microscopy. So electron microscopy I do not know much about. So unfortunately I do not work in that imaging data. Yeah.
Okay, thank you.
Yeah. It's what I understood from the literature is that it's much more complicated. So we have enough problems in other imaging modalities. So I have not worked on the EM, but I mean it's very useful. I mean what I see from the literature is that it's a much more complicated problem. But again, deep learning models is what I see in that world as well. They're using for classifying different regions in electron microscopy data. Yeah.
Thank you.
Mm.
Any queries, participants?
Right.
Okay.
Sir?
Yes, ma'am.
Yeah.
Okay. Okay.
It's my great pleasure for me to deliver the vote of thanks. On behalf of the Department of Biomedical Engineering, PSNE College of Engineering Technology, I would like to thank Mr. Surya Prasad, Assistant Professor, Division of Biomedical Informatics, Cincinnati Children's Hospital, Medical Center, Cincinnati, who gave us a very nice talk on today's first session. Thank you, sir. I'm sure that the participants would be highly benefited from his talk. Thank you, sir.
Yeah, thank you, and please feel free to reach out to me if you have any questions. I sent my email ID in the chat. Yeah.
Yes, sir. We will make use of it, sir.
Thank you. Thank you.
Thank you very much, sir. Thank you, sir.
Thank you. Bye. Have a good
Thank
day.
you,
Thank
sir.
you. Thank you, everyone. Thank you.
Sir.
Bye. Bye.
Bye, sir. participants. I will meet you on the next session at 11:30 a.m. Okay. Sundara ma'am.
Yes, ma'am.
Okay. Venkatraman sir. No,
Venkatraman sir, no, he still didn't join, ma'am.
okay.
Good morning, sir. Sir, you can share your screen now, sir.
Yes. Ma'am, am I audible?
Yes, ma'am, you're audible, ma'am.
Okay, thank you. Good morning, all. I have great pleasure to introduce the guest speaker of this session, Dr. K. Venkatraman, the Technical Manager, Biovision Systems, Chennai. A budding professional, started his career as biomedical engineer since 10 years. He is a doctorate in the field of medical imaging and instrumentation after his postgraduate in medical electronics and MBA in hospital management. He has been associated with Sigma Meditech India Private Limited and AMS Solution in the past years as biomedical engineer. He has also served as an assistant professor in Bharat Institute of Higher Education and Research for 3 years. He has also handled various government hospital-based equipment maintenance projects in Tamil Nadu and Chhattisgarh. Presently, he is also serving as consultant for equipment maintenance at Ecom Foundation and NGO at Chennai, and is a member of Board of Studies for various prestigious universities. He is also an adjunct faculty for various colleges and is training numerous students of biomedical engineering across India. With this small intro, I hand over this session to Dr. Venkatraman. Welcome you, sir.
Thank you, madam, for the introduction that you had given.
Thank you.
So this session, I'm having a briefing or a basic introduction about a different device which most of us would be knowing or may not be knowing also. It is C-arm. C-arm, an inevitable surgical need. So when we talk about medical devices, we basically classify medical devices into three categories. As I say everywhere, it is diagnostic device, therapeutic device, and supportive devices. Supportive devices are few devices like OT light, OT table, ICU cot and all, which neither do therapy nor do monitoring. So when I come to few devices like X-ray, they are commonly used as a diagnostic device based on which therapy is done. So C-arm is one such device which comes under the radiological family. So it is something like an X-ray which we will be understanding in the upcoming slide. So basically what is C-arm? C-arm is a medical imaging device that is based on X-ray technology for use in operation theaters OT. The name is derived from the C-shaped arm. As we say, it is C-arm. You have a C-like structure, so it is called as C-arm. There is something also called as O-arm. O-arm will be like a CT scan gantry. CT scan gantry maari irukku, O maari irukku. Nadula, you can place the table and you can perform your surgery. That is O-arm. So what we are going to see is about C-arm, which has an X-ray source and X-ray detector in the both the ends of this C. Specialists in the field of various surgery like orthopedics has a very common use, and vascular cardiology has a very common use of C-arms. These produce high resolution X-ray images for real-time imaging so that we can monitor the progress of the surgery at any point of time. For another basic understanding, we can simplify as a C-arm is used during a surgery. For example, when will we post a surgery? For example, or ortho case, we are going to do a ortho surgery. For example, you are going to implant something in your bone, a plate in your bone, for example, say. How does a doctor conclude this? By taking an X-ray, not sufficient. They go for a CT scan. From the CT scan, they infer that some artificial implant or external implant needs to be placed inside the bone. Okay, so they have identified using a CT scan or an X-ray. So this is pre-operative imaging, which is obtained from an X-ray or CT. But during my surgery, for example, I'm placing this metal, I'm placing this plate into the patient, for example, say, if such is the case, I will not be having a facility to take this X-ray machine there, and I cannot keep exposing the patient to X-ray and I have to watch the film or CR or DR, whatever it is. You have computerized radiography nowadays, where you take and put the cassette in a reader, you will be getting the image in your system, or DR system, you will be getting time to time images. You put a click, within 2 or 3 seconds, you will be getting the image in your laptop. Yes, that is all possible. But taking a laptop and a cassette to the operation theater may not be advisable, because how will you sterilize that? How will it be autoclaved? These are shortcomings of a X-ray radiography, especially digital radiography. So for these purposes, we can go with C-arm during surgery. So during surgery, I say that I'm going to implant in this particular place. How do I know that I'm implanting only there? So I can take a video kind of frames, continuous frames I can also take, and I can review all these images to see the positioning. So during surgery, I take a C-arm to ensure that the fixation is being done in the proper anatomical site. And after the surgery or in between the surgery, once the fixation is over, you take an image again and you will have to re-verify whether it is properly placed, whether the anatomical structures are all fine. Physiologically, will it be okay? All these things has to be verified after fixation. So you take a C-arm image after final fixation. Suppose you will have to revise it, it's not proper, you have to redo, you have to refix, take and modify something. If such is the case, you go for a refixation and then you take the image if required. Once after the everything is over, you put the suture, close the surgery will be over, and the patient will be coming out of the surgery to ICU or ward or whatever, after which you will have to retake an X-ray or C-arm to ensure it is all proper. Okay, so a C-arm is a real-time X-ray. You can understand that way. Instead of taking an X-ray, watching the film or viewing that in a system on a laptop, you can simultaneously see in your monitor, and you can also perform all the image processing operations in this image which is obtained in your monitor. So initially, first C-arm was developed or was implemented into practice in the year 1955. Just imagine, from the year 1955, what are the evolutions that it would have suffered all these 50 years. So the mechanism basically here of the working of a C-arm is, I'm not going in depth with the instrumentation, because most of us will be knowing when we talk about imaging, X-ray is a very important source or important modality of imaging, and most of us here would be knowing how X-ray imaging works, how a normal X-ray works with soft and hard X-rays, the bones, how are they being captured, a thick tissue, how is it being captured? I'm sure you will be knowing that. So having this in my idea, I am proceeding this further. A C-arm comprises of an X-ray source generator and an image intensifier or a flat panel detector. Even if we use flat panel, We are used to calling it as II. We commonly tell as II, which is image intensifier. It is very common term which we use. Any technician also uses the same term. So as I said, you have a source at one side and detector at one side. The source here is X-ray source or generator. The detector which we use here is image intensifier. The C-shaped connecting element allows the movement horizontally, vertically around the swivel. We will be seeing the degrees of freedom, how far you can rotate or what are the angles with which you can take. By seeing that, you will really be able to appreciate the innovation of a CM. We will be seeing that also later. So you see is a connecting element which can be used to move these X-ray source and detector parallelly. It will be oppositely posed so that whenever you move the source, you don't have to position the detector. Automatically, the detector will also come to its own position. There is a greatest advantage. That's why we use this in theater, because when we use for radiography, you have to position the cassette every time, which is practically not possible. You cannot lift the patient, position the cassette downward. That cannot be done so easily, right? So that is the greatest advantage of a CM, is that your detector moves along with the source, just like a CT. As your source moves, the detector also moves automatically. The generator here emits X-rays to penetrate the patient body. The detector converts this X-rays into visible image. How? We will be seeing it in detail for image intensifier and flat panel detectors in upcoming slides, so that the doctor can then identify and check the anatomical details on the image, such as blood vessel, bone, kidney, everything, whatever it is. You can just simply start your analysis or monitoring, diagnosis using your C arm. So this is the basic introduction of the mechanism. So this is how a C arm will be. So whenever we use an image intensifier, that is going to convert your analog to digital, and then it is being displayed in a monitor. A monitor is basically analog monitor which are being used. These are now not in practice actually, but still the people who had bought it in earlier days are still using the same C arm with image intensifiers. Nowadays the technology has greatly improvised that we are using only flat panel detectors most of the times instead of a CRT monitor. So X-ray source, patient, comes to the image intensifier, and just above this image intensifier will be having a camera unit. By before end of the session, we will be able to open a machine of using this image intensifier. There we can see what is what in detail, which will be handled by Mr. Zahir Hussain sir, our CEO. And analog to digital converter, there you do the image processing and you store this image. That is the greatest advantage. Once the surgery is over, you can write in a CD or you can do it in a pen drive and you can give to the patient to see how the implant has changed their anatomical structure. Initially, how it was, how the procedure was going, how the implant was done, and how it has changed the structure right now. Everything could be viewed as a video by playing these frames in a particular sequence using a monitor. So when we come to flat panel detector, it's a digitized flat panel detector, just like any other digital X-ray system. So it will be detecting the X-rays and it is given to image processing and storage and displayed using a LCD monitor. So coming to evolution, what exactly is this CM, or who is the parent of the CM, or what is the after CM evolution, what has happened after CM, everything can be understood here. So the first emerged was a basic fixed radiography, which is a normal analog radiography, where it's a conventional radiography where we use a film, go to the dark room, watch the film, and then you can get the image. This is a conventional radiography. Then came computerized radiography, where the cassette was readable. You did not have to watch the cassette. Instead, you can read the cassette in the system and you can reuse the same cassette. You don't have to change the film every time. So that was your computerized radiography. I suppose you will be knowing this. And with this idea, I proceed further with digital radiography. The difference between CR and DR is that in CR, you have to take the cassette, put into the cassette reader, and then the image will be obtained in the system. But in DR, you can place the cassette beneath the patient, expose, and immediately within few seconds, you'll be getting the image in your system. So it is very spontaneous. So most commonly used in veterinary applications also, because it will be very hard for you to position the animals. So once you position, you can take multiple clicks also when we go with the DR. And whenever we are using these kind of radiological equipments, please be informed that we will have to get a certification from AERB. Atomic Emission Regulatory Board will have to certify that your equipment is okay, for which you will have to submit a certificate to them called as Quality Assurance Certificate. These are additional details which we should be knowing. So when I say it is quality assurance certificate, you will be checking the emission leakage everywhere. When you use a C-arm, just nearby that X-ray tank, at a particular distance from that X-ray tank. So all these things is being given by AER. You have a tolerable limit. If your machine is not within the tolerable limit, then you will not be certified and you're not supposed to use that machine. Okay. So to ensure this, you have something called as quality assurance certification, which is being done by AERB approved boards, AERB approved companies. They will be doing and they will be certifying you. With this certificate, you can get an approval from AERB. It's just an online portal, free of cost only. You can just apply and you can get it certified, which is valid for 5 years. After 5 years, you have to redo the same procedure to ensure that radiological emissions are being in control. This is a very important thing which we will be discussing even in future slides. Radiological emission is greatly controlled by the board, central government board, AERB. So then it is C-arm, which is an X-ray only, but a portable X-ray where the detector and the source both will move together. Here, if you can see, this is the X-ray tank, X-ray source, and this is the detector or image intensifier. Even an image intensifier, there are various sizes. So then it is CT scan, which is like an O-arm, where the source and detector will be moving throughout the O actually to get a cross-sectional view, and it can be reconstructed. This is a cath lab. I'm not sure if you will be knowing what a cath lab is. Cath lab is referred to as catheterization laboratory, which is commonly used in cardiology. This is a C-arm. If you can see, cath lab is also a C-arm. See this. This is the C-arm. Okay. So cath lab is also a C-arm, but it can move through the theater. It can be moved anywhere. When I say it is a C-arm, I can slide the C. Okay. But I cannot move it very easily. You have to put this portable, you have wheel, you have to change it somewhere like that. But here it is remote operated, where you can move it like anything. A cath lab is a hanging C-arm, ceiling hanging C-arm kind of thing. I think you can visualize what it is. So it is a ceiling hanging C-arm which can move around throughout. Up to my knowledge, cath lab is the very higher end equipment I know in the medical field. CT, MRI, it's nothing in front of a cath lab. Cath lab is a very higher end equipment which can be installed and which can be seen in only cardiological specialty hospitals where they post very high critical cases also. So cath lab is also a C-arm, which is ceiling hanged C-arm, which can move around the railing which is put in the ceiling. So as I said, there are two types of detectors. One is analog image intensifiers and another one is digital flat panel detectors. So when I say it is analog image intensifiers, how does it work? The X-ray will strike a fluorescent surface after being attenuated by the different degrees through the patient body. The X-ray passes through the patient body and it will hit the fluorescent surface, and that fluorescent surface will glow based on the input radiation strength. If the X-ray is strong, then the brightness will be greater. So the phosphorus screen which we use will be fluoring or glowing based on the input X-ray. And just behind that, you will be having an image or a camera which can capture this phosphor image. That's all. Over. And that image is being processed with your digital imaging processing system, and then stored in a memory and passed on to the monitor. Only that much. I think it should be very clear. So I'm not, you will be knowing basically about these camera and image processing systems, I suppose. So due to the curved surface of the tube, the accuracy of the image diminishes over the edges. So for this overcoming, we came with something called as digital flat panel detector to avoid these kind of edge or distortion at the edges, loss of information at the edges. So these are modern flat panel technologies, just like a digital radiography. We have flat panel technology here also, except that it is directly connected to the monitor. It is not movable. You cannot remove the detector and you cannot move the detector. So the intensity of the incoming X-rays is converted into a digital value by these photo detectors, these radio detectors. And this is then given to your digital processing system. So first time this digital system was innovated in the year or implemented in the year 2006. So if you can see 1955 to 2006, it took almost 50 years for us to digitize or to improvise or to overcome these distortions or erosions which were occurring in the analog mode of technology. So coming to the positioning of C-arm, there are various things which needs to be taken into consideration when we use a C-arm or any other radiological device. The thing is, the patient as well as the technician must be exposed to very less radiation. That is the basic idea, because radiation has its own harmful effects, as you will be knowing. So to overcome this, we will have to ensure that both the patient as well as the technician are being exposed to minimum radiations. So the first thing is you will have to position the C-arm such that the radiation is minimized both to the patient as well as to the user. So for that, we have to ensure that the patient should be closer to II instead of X-ray, number one. And number two is that always your X-ray tube will go down. Image intensifier will come up. Why? Because you can see in this image, it will be very clear. If the X-ray tube is down, you can see these are the X-rays. These shaded regions are the X-rays which are penetrating within the lower region of the table. Here, if you are keeping the X-ray tube upper side, you can see that the exposure to the patient is also high. The exposure to the technician is also high. So it is always advisable to use X-ray below the table. I suppose you must be getting a doubt here. How, see, I'm using a normal OT table. OT table, which is a rigid surface throughout. I am using an X-ray source below the table. Will this table also appear in my X-ray image? So this will be the greatest question. So no, this table will not appear in the X-ray image. Why? Because you cannot take an X-ray with a normal OT table. To take an X-ray, you need C-arm compatible OT table where all these mattress just below the mattress will be having that base, no, where the patient lies down. That is made of a special acrylic which will allow X-rays, which will not scatter or which will not reflect X-rays. It will simply pass the X-rays through it. So that is called a C-arm compatible table, which is a mandatory tool to be used along with C-arm. Another way to reduce the exposure to the patient. Suppose my area of interest is less. I am focused with a particular area of interest and I need a high quality image. If such is the case, I have something called as a collimator, which is also used in any other radiological device. Even in DR or X-ray, we use this something called as collimator, which is used to narrow down, which is used to focus the X-ray, which we expose to the patient. So when I have an open beam, you can see that the scattering is also high, so the image degradation will also be greater. When it is collimated, there are two advantages here. One, the image quality is also improvised. The patient is also given with a lesser radiation. In turn, the technician will also be given with a lesser radiation. This is the idea of using a collimator. How can we move the C-arm? I was telling, no, you can move the C any way as you wish. How can it be moved? If you can see here, one thing is this way. I think you can understand. You can swivel, na? You can rotate this like a CT. Just imagine if it is a CT, how will it rotate this way, right? So up to this degree, you can just rotate this way. This is swivel. Vertical height, increase, decrease. Increase, decrease the height based on the patient and table position. Horizontal, front and back motion. Can front and back motion. And rotation is this way. This way. Try to understand. Not like CT. Instead, it comes through this. Slide this. For example, the intensifier comes here in this area, and the source will be opposite to this area, like this. So up to 190 degrees you can rotate this way. So that is rotation. So these are the 4 degrees of freedom which is given in a C arm basically. So with which you can cover almost all the angles with which you can cover a CT. The CT எடுக்கும்போது you can cover all the angles. Yeah, same way you can cover all the angles when you use a C arm based on the positioning for which you will be needing an expertise. Not everyone can position this so easily because to view a particular tissue in a particular angle, you need to position it properly. That is very, very important, which can be done by any technician. So params of concern, you will have to reduce the amount of exposure of exposure of the radiation, adjust the distance so that both the patient as well as the technician are getting lesser radiations. Ideally, the patient should be placed 12 to 15 inches away from the source. So we are placing the patient as close as possible to the image intensifier, and only whoever is authorized should be allowed inside the room. And when the procedure is performed, the technician has to be near the image intensifier, not near the source. Suppose you are using parallelly, for example, the example which we saw was the X-ray tube was below the table. Now, if both are parallel, for example, both are upwards, you are putting the C horizontally, then you will have to be at the side of image intensifier and you will have to be away to the patient. And remember, the whole operation theater where we use CM should be lead shielded to avoid exposure outside the theater. And we have our own accessories to protect ourselves, like lead aprons, thyroid shield, and gonad shield to protect, to avoid the exposure of X-ray. And we will be having radiation badges and dosimeters. The radiation badges should be worn, and we have to ensure that we are not exposed to more than 20 sieverts per year actually. So if such is the case, you are getting exposed to that, you will have to go for a vacation for a few while and then come back, because radiation has its own harmful effects and it is also carcinogenic. So I said that the C-arm image intensifier is of various diameters. So based on the diameters, again the patient dosage is also controlled. Higher the diameter, greater is the field of view. High resolution you will be able to obtain from that. The intensifier diameter is greater, lesser is the patient dose, and that is being shown in this presentation. So as far as the instrumentation part of it is concerned, I'm not going to focus much in the instrumentation part of it because we'll be seeing in the practical demo. So with this, I conclude my theory. Any doubts you can please ask now. Any doubts? Any doubts? Shall we proceed, or
Participants, do you have any doubt?
if not, I shall proceed.
Okay, sir, proceed, sir.
Yeah, okay. actually see this is the permitted level by AERB. So AERB recommends that the leakage should not be more than this. The radiation should not be more than this. This is a cath lab actually, which is a hanging C-arm, most commonly used in cardiac cases, catheterization laboratory, used for cardiac cases. So before starting the practical session, I would just like to give a brief introduction about our CEO who is going to handle this practical session. It's a pre-recorded video. Mr. M. Jahir Hussain is a technical expert with over 30 years of experience in healthcare sector. He has served as Director Technical for Sigma Meditech India Private Limited, Chennai, has handled various medical equipments and catered to the needs of all specialties of medicine. Presently, he is also heading the technical operations at AMS Solutions INC Chennai, which deals with a wide range of medical equipment in general segment, anesthesia, critical care, and cardiothoracic to be more particular. His core competence is high level of technical skill, strategic planning, new product identification with all India dealer network operations and management. Mr. Jahir Hussain has gained a unique identification in the medical industry of Indian origin, who commands a high regard and respect for his professional ethics, dedication, and devotion. His strength is commitment in offering 24/7 services and customer support to all the healthcare professional across India. So with this brief introduction, I'm proceeding with the presentation of opening and instrumentation part of C-arm, after which we will be having a briefing about high flow oxygen therapy, which was commonly used for this COVID treatment.
Good afternoon, everyone. Today we are going to take about the CM classes. First thing, what is CM? Why it names CM? Because it is the shape is a C type of shape. That's reason it is called a CM. Actually, this is basically it is again the X-ray family. What is the difference between X-ray and CM means? In X-ray, it will be just fixed in one place. You can take the object or anatomy in between that. You can take the images. Images means it will come through the film. Or nowadays, there are a lot of new technologies like computer radiography, CR system, and DR system. So many new techniques has come. Okay, so in the earlier the CM was invented in 1955. It is they started with ortho. Suppose you want to do any cases in the theater, like bone, you want to implant anything, you want to keep some screws, or you want to put a plate and all that. So after fixing that in the surgery, during surgery, after fixed this type of ortho implants, they want to confirm with the X-ray whether it is fitted properly or not. Not only ortho, the CM is using in different you can use it for in urology, you can use it in cardiology. Of course, cardiology means again the same CM in cath lab. You heard about the cath lab. Cath lab also similar setup, but it is very less than kV. In that cath lab they will use more kV and some other different type of images they will take it. And also it got some memory unit. It stored the image. Suppose if they are taking during the surgery, they will take a shoot. First they will take a picture and they will store in the memory unit. Okay. Before that, I will inform you. There are two parts in this CM. One is C-arm. This is normally kept in the theaters. OT they will use this CM, and this is a memory unit. So whatever you are taking the image, that will be stored in the memory. So later on, after the surgery, you can recall the memory. You can go through that. So what they fixed or not, like that. Okay. CM again is after the CM. Suppose in X-ray, in this part there are two parts. One is the X-ray. This is the X-ray tank. This one is the image intensifier. See, now this is a old type of IA. So current now old type means this and all that CRT. You heard the old TV and all that, then they use the cathode ray like that, they use this. So once X-ray is generated by tube, so it is passing through the object means either anatomy, so again it will be captured by the IA, again it is converted into the imaging. Understood? So this is IA. Anyway, we will open and see that IA, how it is working and all that. I will tell you later. Before that, just I am giving you the idea what is what. Okay, again, memory. Memory actually in the memory unit there are two monitor will be there. One monitor is taking the live image. Other monitor you can store the previous one. So that the doctor can see that what is the difference between the object. Okay, and of course you can. This is actually memory unit. In that memory unit, you can store up to 100 images. Suppose you want to store more images, then you can put some computer and a graphing card, so you can store it also. This key can operate through the console. In the console, here you can see that kV and MA, how much kV you need and how much MA you need. Okay, and so up and down. Suppose they have the inside the machine, they have the motor. So the motor will move the up and downs of the C, and you can also rotate 180 degrees. 180 degrees you can move it. Okay. So here we are setting about 58 kV. So you want to reduce. This kV also depends upon the object. Suppose I'm going to take my hand, so I can keep about 40 to 50. See, once I set the kVA, so this is actually foot switch. Either you can operate through this foot switch, or you can have the exposure switch will be available in the system also. So normally this foot switch will be kept in the near the surgeon. So once you want to see the image, so in between that they will put a object, means the patient will be there in between that, okay, and in between the patient will be there, and the table also sometimes they will use a special type of tables. So all the OT table cannot use for the CM. So it got the CM compatible table is available in the market. You can use that one. Okay. Just now I kept my hand up in between that X-ray and IA. See the monitor. Now actually I kept 40 kVA, but my image is not clear, so I need a more kVA to expose. So I'm increasing this kVA around 50 above. Now you see the quality. See, so I can see my hand. These are the bones, what are the bones available, and so like that you can expose like a hand, or you can take some arm. Spine. Okay, this is spine of patient. So here you can see all the L1, L2, L3 like that. So that and all that doctor will know that. Just I am showing how to take the spine and how to take the hand and all the imaging that I am doing. Okay. So after that, after the expose, either you want to, you can adjust like a post processing. Here actually mic. Just I showed you what is CM, how to expose, how to keep the KV and MA, and how long I can shoot, everything just I explained to you. Anyway, what we'll do, we'll open the CM and we'll explain some more techniques. Okay. In the market, there are a lot of companies, competitors in the market. Like the major companies like Philips, GE, Siemens, they are all pioneer. Okay, and of course they are all importing from like the US and UK, different countries. And in India also, in India we are making concept. They are also making some local companies are available like Challenges, RMS. So what they will do, they will assemble all the X-ray tube and other part. They will import only the tube. Tube is from like a Toshiba and some Philips. They have the different type of IAs are available in the market. Okay. So now we are opening on CM. We are opening on CM. First they are removing. This is IA part. Image intensifier. So you can see that. Actually, you can see a lot of so many components inside the tube. Tube will be, this is a tube. Tube, this is a tube. Tube is like so inside the tube will be there. This is a camera, okay, and you can see some. This is a power supply, high voltage power supply. So this will generate more than 25 kV. So this only absorb the once the X-ray is emitting, it will hit the IA tube. So the IA tube inside the phosphorus will be take a, it will come out from the tube. So that will be observed by the high kVA voltages. So normally it will be about 18 to 25 kVA potential inside. So before opening that, you make sure all the safety features, you have to remove the power cord and other things, then only you have to open it. This is a camera. This camera can also remove from the, see here the tube, inside the tube will be there. So the image will come this lens. This lens will be connected with the camera. So camera will observe the image, and after that it will process all the digital process and everything. Then the signal will be transmitted through the video cable to the console. Okay. This is the camera. So here you can see that this high voltage power supply. This is all a circuit, electronic circuit, which will deliver the voltage. Now we will open this X-ray section. See, this is the X-ray generator. Okay, from this is again it's called a collimeter. Suppose you want to take the image. It's called a region of interest. So a particular point you want to focus and then take it. You need the collimeter. So this collimeter also you can adjust through the keyboard. How much shape you want, like that. So if you open this, then you can see the tube. It's a tube. So the tube is filled up with the oil. Okay, inside the tube there are oil will be there. Removing the highway tube. These are our radiography, radio imaging system, and also it should be this license will be provided by the AERB. AERB means Atomic Energy Regulation Board. So they have to issue this license. Okay, because in there they will have some norms how to, suppose you are going to keep this CM inside the theater, or you are going to keep the X-ray in the different place. So they have some norms. What is that norms? You have the security, safety and security. Okay, so they will construct the wall thickness should be like this. After the wall, you have to put some lead glass and lead tubings. Okay, that and all that be decided by the AERB. So once if you apply the AERB, once you get the equipment, you have to apply for the AERB to the hospital. So AERB will inspect and they will certify whether your CM or your X-ray system all are safe conditions. So you can, because it should not harm to other patients. Okay. So here the tube, it's like a glass tube. So here only the X-ray will hit, and all the phosphorus inside it is coated with the phosphorus, green phosphorus. Okay. Now we'll first I'll remove the camera and then we will connect it. So only one connector, and this is video cable. So now I separated the camera. Okay. So now I am removing some cable from the C. Okay, isolated. So now the tube is isolated from the C. So while taking the IA, because it is like a counterbalancing, you see, if I am removing that, before that I have to put a lock. This is the lock. I will close it. Now it will lock. So before opening the IA, you have to take a extra care, and you have to follow the company rules and regulations. How to open, what are the safety features you should take, everything. You have to read the manual before opening the IA. See here, you can see that this is the power supply. I told this will generate the, you are applying the 48 volts, then it's inside the high voltage converter will be there, that will increase the voltage like a 25 kV. This will go to the anode of the IA mentioned things, right? Okay, and also the board is also very costly, and you cannot you can remove the power supply, but you cannot repair it, because this power supply was completely isolated with some kind of special material, so that there is no, because it's a high voltage, we should not arc to other boards or to the tube or something. Okay, so that's the reason the power supply was kept inside the tube. Okay, so here only after the 230 volt will be converted into multiple voltages like 5 volt, 12 volt, 24 volts, 48 volts, like that they will convert it into. And also it's this high voltage also creating from here side only.
Any doubts related to C-arm? If not, we can proceed with our next session of high flow oxygen therapy, which is a respiratory therapeutic device. Any doubts in C-arm? If not, we can proceed with our next presentation.
Sir,
Yes, ma'am.
sir, is there any possibility to detect how much of magnetic that is radiation is there
Okay.
on the patient or surrounding the patients?
Okay, nice question. Actually, as far as the C-arm is concerned, we will not be getting any magnetic radiation. Instead, we will be getting ionizing radiation. To monitor this, what we do is we have to perform quality assurance. We call it as QA test for any radiological device. What we do in this QA test is that we will be analyzing the amount of radiation at every diameters, outside the theater, inside the theater, within particular range or diameter of the theater. We will be analyzing the amount of radiation that is being exposed using a dosimeter, and this should be done only by a AERB approved lab, after which you have to submit this report to AERB, and then they will certify you saying that you can use this C-arm. Until then, you are not supposed to use this device. If you are using this device without such permission, then they may even seal your hospital. So it is a very high, that's a good question actually. So for any ionizing radiation for that matters, you have to test the amount of radiation using dosimeters. You have to get it quality assured from a AERB lab. submit this report to AERB and get their approval through LORA, and then only you can proceed using this device. If not, you're not supposed to use this device. Hope this answers your question.
Yes, sir. Yes, sir. Yes, sir. Thank you, sir.
So I think I can proceed with my next presentation
Yes,
of
sir.
high
Yes.
flow oxygen therapy. Sure. Before
Yes, sir.
getting into my presentation of high flow oxygen, I would just show you a small video about what has happened during this COVID situations actually. So after which we will proceed with our presentation.
A day after Central Gujarat's biggest public hospital witnessed a fire inside the intensive care unit of the COVID hospital, CCTV footage of the incident emerged on Wednesday, revealing that the fire was not from a short circuit, but because of a ventilator placed inside the ICU-1 unit located on the first floor of the SSG building. The footage, which was widely in circulation, clearly shows that there was a kind of blast inside the ventilator, after which the fire started, resulting in panic among the staff and patients present inside the ICU. The CCTV footage shows how the staffers bravely rescued the patients one by one, even as fire had started spreading inside the unit. It is a possibility that the ventilator caught fire due to heating, but the exact reason can be known only after the forensic investigation is done. Forensic experts' help has been sought for the same. The Casualty or Trauma Center of the SSG hospital, which houses the COVID-19 hospital, had provisional no objection certificate of Vadodara Fire and Emergency Services, as the firefighting equipment was intact and in working conditions. Officials maintained that a 4-member committee is probing the reasons behind the fire.
So the basic idea behind this video is that we need to know the safety parameters that needs to be taken into consideration when we are planning to design any device. So with this basic idea, I'm proceeding with my high flow oxygen therapy device. Further to the understanding about low flow oxygen therapies, which is being very commonly used in most of the hospitals, now we can go with something called as high flow oxygen therapy, which was existent even years before, but which has popularized or which have become very familiar to us or even to non-medicals in this pandemic situation of COVID-19 actually. So there is something called as high flow oxygen therapy, which has been focused nowadays for treatment of respiratory disorders basically. To understand what it is, especially for COVID-19 conditions, the patients are suffering lung infection, respiratory tract infection, alveolar infection, and hence the saturation of oxygen, the amount of oxygen that is being diffused into their blood is greatly reduced. That is the first problem. So they have suggested few remedy. The WHO has also suggested some recommendations like prevalence of hypoxic respiratory failure is greater for COVID-19 conditions. So they suffer hypoxia and shallow breathing. So if it is shallow breathing, the respiratory rate increases, but efficiency of breathing will be reduced. So the rate is exceeding 30 breaths per minute and the saturation is reduced to less than 93. Suppose if it is reducing 90, if it is reducing less than 90, highest concern should be taken care to see what exactly is happening to the patient. But how do I check the saturation for that? There is something called as a pulse oximeter, which you actually already know. But this has become a home care device nowadays, which is called as a fingertip pulse oximeter. A very low cost device which is easily available everywhere. A fingertip pulse oximeter, which you just put in your fingertip, you get the saturation and pulse rate. Understand? So this fingertip pulse oximeter works by the principle of photo gate. It has a LED LDR pair by Beer-Lambert's law, which LED emits some light and the received light intensity will vary based on the oxygen concentration in your blood. So this LED LDR pair ensures that SpO2 and pulse rate value is obtained from the device which is called as fingertip pulse oximeter. Supposing the oxygen percentage is lesser, SpO2 is lesser in this fingertip pulse oximeter, then we have to go with administering the patient with supplement of oxygen. You have to supply the patient with additional oxygen to ensure that his saturation is properly maintained. But how do I ensure the supplement of oxygen? How do I deliver oxygen? By means of various modalities which are present already. You have a cylinder from which you can put a regulator and you can directly give oxygen to the patient, number one. You can use a oxygen concentrator, which is a device which generates or produces oxygen from the atmosphere, number two. But the basic disadvantage of these two machines is that these two provide oxygen at a lower flow rate, lesser flow rate. Only up to 10 lpm a concentrator can give, up to 15 lpm a cylinder can give. But suppose this is not sufficient for the patient actually needs much higher levels of oxygen. In that case, we go with a device which is called as a high flow oxygen therapy. So to administer this high flow oxygen therapy, first we will understand the low flow oxygen therapy method, which we would have already been exposed to. An oxygen concentrator extracts the air, compresses the air from the atmosphere, converts it into oxygen using zeolite crystals. That is done by an oxygen concentrator. So oxygen concentrator converts. Please understand the difference between HFNC, that is high flow oxygen therapy, and oxygen concentrator. Both are totally different machines. So when I say it is an oxygen concentrator, it generates oxygen. It is an oxygen generator. But HFOT, oxygen therapy, is not a concentrator or a generator of oxygen. So in concentrator, you can see that the nitrogen is being absorbed and desorbed by the zeolite crystals. But these kind of chemical reactions are not happening in high flow nasal cannula or high flow oxygen therapy. Instead, in high flow oxygen therapy, you have to externally give oxygen. Remember, it is totally different. HFNC has come as a replacement for ventilator for COVID patients, where you have to supply oxygen, central supply oxygen, high pressure oxygen is required to operate an HFNC. So along with this oxygen, you also combine compressed air and you determine the percentage using a blender. See here you can see flow meter air oxygen blender. The air oxygen blender is used to set the fraction of inspired oxygen, that is FiO2 level, is being set using a blender here. So after setting that, from the blender it comes into the flow meter. So what does a flow meter do here? In the flow meter you can actually set the value of flow up to 80 lpm, from 0 to 60 in few machines, from 0 to 80 in few machines. So you can set the liters per minute from 0 to 60 or 80 based on the patient condition. If the patient is in need of higher amount of oxygen, then you can go for a higher rate. Up to even 60 lpm or 70 lpm can be given to the patient. If not, if the patient is required with only small amount of oxygen, in that case you can administer with 30 or 35 lpm itself. But as I already said, you are not supposed to give dry air into the patient. It will cause pulmonary inflammation in the airway tract. To avoid this, you will have to humidify the air that is being taken in by the patient. So how do we humidify the air? In oxygen concentrator, you are using a jar with water which was adding up some humidity. So here we will be using a real-time humidifier, a servo humidifier, to maintain the temperature with which the air enters into the patient's lungs. So for example, if the air is to be entered into the patient lung with a 37 degrees Celsius, for example, say, then that 37 degree is being achieved by a servo humidifier. But how? You have to warm the water. You are trying to warm up the water, heat the water, and you create water vapors, and the air that is entering into the patient takes this water vapor and it reaches the patient's nose. You can see here from the flow meter it comes to a jar called as an active humidifier. From the jar it goes to the patient nose, right? So there you have to check the temperature with which it is entering into the patient nose. Suppose there is a drop in temperature, then what you do, you will increase the heat. Suppose the temperature is higher in the patient end, then you have to reduce the heater current. So this is called as a servo humidifier, where you alter the temperature with which the patient inspires the air. It is also a very important tool. The major challenge which we face in design of high flow oxygen is that we will not be able to maintain this temperature 32 or 34 or 37 or whatever be it. We will not be able to maintain this temperature. Why? Because the gas flow is very high. The flow rate is very high. At this point, I may not be able to maintain the temperature. You have a cup of coffee, you blow it slowly, then the temperature will be maintained for a longer time. You blow it with a higher rate, higher flow. What happens? It gets cooled off immediately. Yeah. So that is the principle here also. So as the flow is greater, the heating efficiency should also be high. And that is not able to be met out by us because you may be using 30 LPM or 20 LPM or 80 LPM. For all these ranges of flows, your humidifier should be adaptive. So that is a greater challenge which we were facing, but of course, technology is a boon to overcome all these shortcomings. But still, it is a parameter which needs to be taken care of when we are talking about high flow oxygen therapy. So what is the difference between the high flow and the low flow is that in low flow, usually when we respire through our nose. But unfortunately, sometimes through our mouth also we respire. Right? So whatever you give it through your nose is not only that enter into your lung, something from your mouth also enters into your lung. So if you are using low flow, when your mouth is also breathing, what happens? The gas mixes and ultimately the effective percentage of oxygen which is reaching your lung is reduced. From this figure you can understand. But when I use a high flow oxygen, as the flow is higher through my nose, with a higher percentage, the effective fraction of inspired oxygen, the effective FiO2 percentage is greater in a high flow oxygen, so that blood carrying oxyhemoglobin capacity will also be increased, so that the peripheral oxygen SpO2 can also be increased. As more amount of oxygen is infused through your respiratory tract, it is expected to increase your saturation and make you stable. More amount of oxygen is delivered to your tissues. Understand? So this is the basic difference between a low flow oxygen therapy and a high flow oxygen therapy, and this is the need of oxygen therapy. And one more thing which is important is that you have to use a proper cannula dia. Proper sized cannula should be used there. Failing to which there is a loss of efficiency again, because air will be mixed up with the oxygen which you take inside. So the cannula size is very important. The advantage is that it can match the patient demand. If the patient needs more oxygen, you can increase it. So it can match the patient demand, number one. The dead space is greatly reduced because it is a continuous flow. Unlike a ventilator or any other device, you will not give a flow, stop it, give a flow, stop it. No, it is not pulsatile. It is a continuous flow, so there is no dead space. The flow is continuous, the dead space is not there, right? Because there is a continuous flow here. And it provides a positive pressure in the upper airways, which will ease the patient to breathe further. That is an advantage. And as the flow is continuous, the mucus secretion will not get stagnant in your airway. The mucus secretion, even if it is present, it will be flushed off by the high flow oxygen which is being delivered to you. The greatest advantage is that it is a non-invasive mode of treatment therapy. So that is also an advantage of high flow nasal cannula or high flow oxygen therapy. So continuous flow of oxygen washes out the upper airways continuously, and it always acts as a reservoir of oxygen. Whenever patient needs higher amount of oxygen, it always supplements that. And rebreathing CO2 can be avoided. Whatever is expired by the patient will not be rebreathed because it is being flushed off continuously. The flow which is present, the continuous flow which is present will be flushing off the carbon dioxide also continuously. So this is the biggest advantage of HFNC. To summarize about HFNC, it gives heated and humidified gas based on the inspiratory demand of the patient. It increases the residual capacity by maintaining a PEEP with high flow, and it is very light, unlike a CPAP or BiPAP which will pressurize your it will be with a high pressure, a high flow actually, CPAP or BiPAP device. It will pressurize. That is not present in a HFNC. It is milder, it is lighter. With proper oxygen dilution from 21 to 100, which can be set by us and which will wash out the dead space for effective flushing out of CO2, effective removal of carbon dioxide, right? So this is all about a HFNC or a HFOT, which I just wanted to cover in this situation especially. So any doubts in this? The instrumentation is very simple. You have a blender, blender flow meter and humidifier, blender flow meter, humidifier, patient. That is it. Nothing else. You can make an HFNC by your own. Buy a blender, connect a high flow meter, output to proper humidifier. That is the challenge again. Connect it to a proper humidifier, put it to the patient. That's how they give therapy nowadays also in most of the hospitals. So with this I conclude my session about high flow oxygen therapy or high flow nasal cannula. Any doubts? You can please ask me. Any doubts, please?
Any standard range of oxygen in human body?
You mean to say in percentage or LPM, sir?
Percentage that low and high level oxygen level.
Yeah, what we can set is from 21 to 100% of oxygen can be set in a machine, but our lungs are capable of taking whatever oxygen is required from this 21% only. For example, you inhale 21% of oxygen and what you exhale will be 18% of oxygen. So your lungs will be consuming only 4%, out of which all the 4% will be diffused through your body for its
Blood.
own functioning.
Through blood.
Yes, sir. Yes. Software available for this system for HFNC. There is no such software and all. It's all basically hardware based design, controller or processor based only. Most of the time these blenders are all mechanical blenders where you can use mechanical valves or solenoid valves. If at all if you're going for electronic blenders, then you can go for proportionate solenoids. So for any interface kind of thing, for patient interface, you will be having a patient circuit, the cannula, the tubing is only for the patient interface. So machine will be operated with gas system with oxygen and air independently that can be interfaced with the patient through a tubing.
Okay, thank you, ma'am.
Thank you. Yeah, further to this, I have a last video, which is an awareness video, an animation video, basically. I think with that, we can end up the session. Just a few minutes video. Yes, with this, I conclude my session. Any doubts, you can please ask me. I've also posted my mobile number and mail ID in the chat box. Any queries, I'm always open. You can always have a contact with me. Sure. So, anything else? With this, I conclude my session. Thank you. Thank you for listening.
Thank you, sir.
Thank you, ma'am.
Mohan, sir.
Yes, ma'am, I'm here.
One time.
Good morning, participants. It is indeed a great pleasure for me to deliver the vote of thanks. On behalf of the Department of Biomedical Engineering, PSNA College of Engineering and Technology, I would like to thank Dr. Venkatraman, Managing Partner, Biovision Systems, Chennai, and Dr. Jahir Hussain, CEO, Biovision Systems, Chennai, who gave a nice talk on C-arm and high-flow oxygen and necessary inpatient stabilization. I'm sure that the participants would be highly benefited from this talk. Thank you, sir. Thank you so much.
Thank you, sir. Thank you.
Thank you, sir. Thank you very much, sir. Participants, the link for the feedback today morning session has been given in this chat box. So finish it off and we'll meet you in the next afternoon session by 2 o'clock. Thank you.
Ma'am, somebody have raised hand. Shruti ma'am have raised hand.
Ma'am, do you have any clarification, ma'am?
K. Shruti. Madam, do you have any clarification?
No, sir, it's by mistake, sir.
Okay,
Thank you, sir.
Thank you, participants. Thank you. We'll meet again by 2 o'clock.